And the last speaker for today's program is joining us from the US, Professor Earl K. Miller, Massachusetts Institute of Technology, USA. He will be speaking on the topic From Clockwork to Emergence, a new view of the brain. Earl K. Miller is the Pickover Professor of Neuroscience in the Pickover Institute. He received an MA degree in 1987 and PhD in 1990 in psychology and neuroscience from Princeton University. He was awarded an honorary doctorate, Doctor of Science, from Kent State University in 2020. He studies the neural basis of executive brain functions, the ability to carry out goal-directed behavior using complex mental processes. His research topics include working memory, attention, decision-making, and learning. The Miller Lab has shown how categories and concepts are learned, how multifunctional mixed selectivity neurons endow the cortex with computational horsepower and flexibility, and how neural oscillations regulate neural communication and consciousness. This work has been established a foundation upon which to construct more detailed mechanistic accounts of cognition and its dysfunction in diseases such as autism, schizophrenia, and attention deficit disorder. Professor Miller is the recipient of a variety of awards and serves as editor and on the editorial boards of major journals in neuroscience and on international advisory boards. His paper within Jonathan Cohen, Miller and Cohen 2001, which presented a new framework for understanding the prefrontal cortex, ranks 5th of all time in citations in neuroscience. Please help me in welcoming Professor Earl K. Miller.
Thank you very much. Thank you for the opportunity to speak to your conference. I really appreciate it, and I wish I could be there with you. So let me just get started. Okay, you should see my full screen now. Okay, well, hello everybody. Again, thank you for the opportunity to speak to your conference. I'm going to tell you about our lab's work on the neural basis of high-level cognitive functions. So our lab studies executive brain functions. This is the brain functions involved in planning, focusing attention, learning, following rules, the kind of things we associate with high-level thought and action. And we study them by recording electrical signals from the brains of non-human primates. We use computational modeling and analytics to make sense of these electrical signals. And we do so because we want to understand cognition. And that's important. Understanding cognition, understanding the brain is important. But we also want to understand cognitive dysfunction in diseases such as autism, schizophrenia, attention deficit disorder, so that we can open up new paths to treatment. Now, today, I'll be talking about some relatively new technology, new-ish technology. I say new-ish because this is technology that did not exist when I was a graduate student back in the 1980s. We used to focus on the neuroscience in the 20th century, used to focus on the brain's individual parts. What are the functions of individual brain areas? What are the functions of individual neurons? Part of that was because we didn't really understand a lot about the brain back then. We understand more now. Another part of it was technology. Back when I was a graduate student, we could only record from the brain one neuron at a time using single electrodes. But now we have the technology to record signals from hundreds or even millions of neurons simultaneously. And that has led to a new understanding of the brain. It's allowed a new understanding of the brain on the network level, something called its emergent properties. Emergent properties are properties and mechanisms that only emerge when the parts interact in a wider whole. You see properties at this holistic level that can't be reduced to the properties of individual parts. And I'll give you an example of that in just a few minutes. But today I'll be talking about two different types of signals we record from the brain. One is spikes or action potentials. Think of them as like they are the impulses from single neurons. So think of them as like the individual voices of a very large crowd. I'll also be talking about local field potentials. This is the average activity of millions of neurons near each recording electrode. So they're kind of the roar of the nearby crowd. Now, we use these signals, we study these signals in order to understand the role of the prefrontal cortex in top-down or executive control. We know the prefrontal cortex is critical for planning, monitoring, and executing complex goal-directed behavior. That's what executive control is. Our hypothesis was that the prefrontal cortex needs to learn and use the rules of the game. This is called top-down information. The prefrontal cortex needs to learn top-down information and use top-down information. This is knowledge of how the world works. We learn how the world works, how our society works, and we can use that to predict possible goals, goals that are not in the immediate environment, goals that are not right in front of us, and we come up with plans and means to achieve those goals. So top-down information is our knowledge, interpretations, and expectations of the world learned by experience. And in contrast to that is bottom-up information. Those are the raw sensory inputs flooding into your brain. Now, about 20 years ago or so, actually 25 years ago or so, we decided to test this hypothesis by determining whether the prefrontal cortex really does participate in executive functions by acquiring and learning this top-down information. So we conducted a series of studies where we taught the animals, our monkeys, new top-down information. And then we wanted to test whether the prefrontal cortex really does acquire and use this information. So in one study, we taught our monkeys to categorize a set of computer-generated images as cats versus dogs. There was 3 prototype cats, 3 prototype dogs, and we used a morphing algorithm to morph or blend the different prototypes. So each of these shows a different morph line. Along this morph line, the shape of this cat gradually became the shape of this dog. Okay, and so on and so forth. This cat gradually became this dog, etc. So what there was a smooth and continuous varying through shape space, but we taught the monkeys that there was a sharp boundary somewhere. In this case, it was a halfway mark between the cats and the dogs. That boundary did not exist in the real world. It was something that was learned by the animals. And what we found after this training is that neurons in the prefrontal cortex began to respond to the category of the stimulus. They responded to all the cats, not individual cats, or they responded to all the dogs, not individual dogs. The neurons separated cats versus dogs and ignored the individual cats and dogs, which is exactly what we trained the animal to do. When Andreas Nieder was in the laboratory, he taught monkeys to recognize the small numbers 1 through 5. And again, we trained the animals so that the exact display we presented to the animal didn't matter. We could present 2, 3, 4, or 5 items, either as circles like you see here, or as squares, or as triangles, or different objects. We taught the animals to recognize the numbers regardless of how we presented those numbers. And we found neurons in the prefrontal cortex that became tuned, that responded to the numbers and not what the individual displays actually looked like in detail. We also taught animals high-level concepts like same versus different, or up versus down. And what we found across all these studies is that the prefrontal cortex absorbs top-down information. After this training, the neurons in the prefrontal cortex throw away the details about individual cats and dogs or individual displays, which was not relevant to the task, but instead it learns the high-level principles and rules needed to solve the task at hand. It learns what the numbers are, or cats versus dogs, or they reflect the concept of same versus different, or up versus down. And then we went on to show that the prefrontal cortex could use this top-down information to to control the flow of activity across the cortex. There's a paper we published 22 years ago, so I'll just briefly summarize it by saying that we found that the prefrontal cortex uses this top-down information, much like an orchestra conductor learns the score. The orchestra conductor has the big picture. It has the score of the piece that the orchestra is playing, and it uses that score to conduct the rest of the orchestra. We think the prefrontal cortex uses this top-down information to control the flow of activity in the rest of the cortex in order to make top-down executive behavior happen. Now, in conducting these studies, we discovered a new type of neuron that hadn't been previously reported, what we called mixed selectivity neurons. Now, back in the 20th century, when we used to think of the brain, we used to think of the brain as kind of like clockwork. Neurons were thought to be relatively simple and specialized. Every neuron was thought to have one function and one function only. This is a traditional selectivity that we thought existed in the brain, only existed in the brain, or the only type of selectivity in the brain back many years ago. And these traditional selectivity neurons, it was thought that they detected single features like edges, or things like color or shape, or they might detect a few simple combinations of a few related features. Like there might be a neuron in your visual cortex that responds to the combination of a red object that's in the shape of a ball rolling across a table, and those neurons, their function is to contribute to the perception of a red rolling ball. Now, these neurons do exist in the brain. There are many neurons of traditional selectivity, but in conducting these studies where we taught the animals all this high level of information, we discovered this new population of neurons that we called mixed selectivity. These are neurons that did not fit this traditional mode. Mixed selectivity neurons were selected for, they activated for complex nonlinear combinations of many features with no obvious relation. These neurons seem to have different functions in different tasks, and the different things they responded to in different contexts seem to have no relationship to one another. They were involved in many different things. As a result, we found that these neurons are multifunctional. You can study one of these mixed selectivity neurons and have the animal perform different tasks, and you'll see this neuron taking on different properties in different tasks. They're multifunctional neurons that can adapt their properties for whatever task the animal is doing in a moment. They participate in many functions, not just one function. But why are these mixed selectivity neurons so important for top-down information? Well, my colleague Stefano Fusi at Columbia University and Mattia Rogati, working with my student Melissa Warden, who's now a professor at Cornell University, they performed computational analysis and modeling to show why these mixed selectivity neurons were so important. And what they do, mixed selectivity neurons add horsepower to the brain. They add computational power that allows for high-level cognition. They're like a neural bazaar where a wide range of information can be combined. Because these neurons can do many different things, it means they have access to a wide range of information. And as a result, the brain can combine new information really quickly to form new rules and concepts. Our brains did not evolve to understand rules like red means stop and green means go. Those are arbitrary things we made up that our brains can now do. And this population of neurons allows these arbitrary, unrelated bits of information to be combined so we could form new rules and concepts. They also make the brain smarter. What these mixed selectivity neurons do is they add a they form a high-dimensional representational space that allows the brain to solve more complex problems than it would with just a relatively simple, linear, low-dimensional representational space. It also increases storage capacity. Back when we used to think the neurons in the brain only had very simple properties, it meant that all the information you needed for a given memory or perception or concept, it all had to fit into a very small population of neurons. But mixed selectivity means that the information is distributed across many, many, many neurons. And what that does is it greatly increases the storage capacity of the brain. Computational models of mixed selectivity can learn a large number of tasks without ever reaching capacity. And for all these reasons, it allows the brain to learn faster because all this information can be combined quickly. And it allows cognitive flexibility for the same reason. Since these population of neurons have so much information available to them, they can help the brain produce flexibility in behavior, our ability to change our mind, change our moments, change our goals, change our plans from moment to moment. But this mixed selectivity requires a new way to think about the brain. Back when we used to think that every neuron had one function and one function only, we used to think that anatomy was destiny. Neurons combine their signals because they're wired together. You have two simple neurons back in visual cortex that respond to things like an edge, a line on a screen, respond to a simple edge, and another neuron that responds to another edge. And these two neurons combine their signals in a third neuron, and that forms a neuron that represents a corner. That's what we used to think. We used to think that the neurons had simple properties, and they combine their signals just because they're wired together. The idea was that every perception, every thought, every action has an anatomically unique network, something we called an ensemble. So here's one ensemble here. We'll call this the blue thought. The idea was that this blue thought here had one separate network that was anatomically interconnected. And here's another thought. We'll call it the red thought on the bottom here. And this thought had another anatomically unique network that was all by itself. You could form associations between these thoughts by creating some extra links, but generally it was thought that every single perception, thought, and action had one network, one ensemble associated with it. But if anatomy is destiny, how does this work? Mixed selectivity means that neurons are not members of one network or one ensemble. Neurons are members of multiple networks or multiple ensembles. So here I've combined our red and blue thoughts into one anatomically overlapping network. Here are the red elements, the neurons that contribute to the red thoughts, the red ensemble, and here are neurons that contribute to the blue thought, the blue ensemble. And there's a population of neurons, the mixed selectivity neurons, these multifunctional neurons that are part of both networks. But if anatomy is destiny, how does this work? Let's say I want to think the red thought. If I want to think the red thought and anatomy was how the brain worked, anatomy only, then if I try to activate the red network, the activity will run through the mixed selectivity multifunctional neurons and start activating the blue network. Now I have both the red and blue networks activated, and my thoughts are a jumble, a mush, a combining of the both thoughts. And mixed selectivity means that neurons are not just members of two networks, they may be members of dozens or even hundreds of networks, hundreds of ensembles. So how do I just activate one ensemble, one network, without activating other networks if mixed selectivity means that neurons, that different ensembles share these common elements? How does that work? Well, one solution that many of us have come to is that neural oscillations solve this problem. Neural oscillations are brain waves. We're probably all familiar with what brain waves are. Here are recordings from the scalp, the EEG recordings from a human. And we know that the brain is highly oscillatory. You see neurons activating together, and that's what oscillations are. There are millions of neurons activating simultaneously and going quiet simultaneously. Activating, going quiet, all together in a synchronized fashion. And that happens from 1 hertz all the way up to 30 hertz or even 100 hertz or more. Your brain does this on multiple scales, multiple frequencies. And what oscillations represent are the large-scale organization of millions of neurons working together. And to help illustrate this, here's a crowd in the stadium on the right here, and the crowd is doing the wave. You can see the wave moving from the right to left on the screen. Now, what this means is that a stadium crowd, a group of strangers who have they're not communicating with one another in a big way. What they're doing is they're following a few simple rules, like stand up when the person on the left of me stands up, and then sit back down immediately. And from those two simple rules, the stadium crowd, this crowd of people, crowd of strangers, can self-organize themselves into something that has a larger structure to it. Now, if a stadium crowd can self-organize using a few simple rules, imagine what your brain can do with that principle. And in fact, that's the exact kind of organization, this large-scale organization, is exactly what is needed for top-down executive control. Executive control, goal-directed behavior, requires getting large numbers of neurons, millions of neurons, on the same page, coordinated and directed towards the function at hand. So executive control requires getting millions of neurons to get their activities synchronized and doing the same thing and directing it towards a goal or participating in a given function. Well, that's the scale at which neural oscillations work. They're the self-organization of millions of neurons working together. Just the scale of organization we need for executive control. So the way to think about this is that neurons that hum together temporarily wire together. Here's these two overlapping red and blue networks, and the red network gets formed, the red ensemble, the red thought happens because the elements that are participating in the red ensemble all synchronize their activity together. And then if you want to think the blue thought, the neurons in the blue network, including the mixed selectivity neurons, they then synchronize their activity together. And now you have two separate ensembles. They may have anatomical connections, but because their rhythms are different, you can form one ensemble versus the other. So neurons that hum together temporarily wire together as they are humming. So what this means is that brain anatomy is not destiny, it's possibility. Think of brain anatomy as more like the road and highway system. Brain anatomy, the road and highway system, just says where traffic, neural signals could go. But your thoughts at any given moment are where the traffic actually is flowing at that moment, which specific streets the traffic is flowing on. And we think these brainwave patterns, this pattern of humming, these resonance patterns of neurons synchronizing their activity, that's what directs the traffic. Now, is there any evidence for this? Of course there is, or I probably wouldn't be talking about it. Back when Tim Bushman was in my laboratory, he's now a professor at Princeton, he conducted a study where he taught monkeys to switch back and forth between two different task rules. The monkeys were looking at an oriented colored bar. It could be a vertical bar or horizontal bar, and the bar could be red or blue. And we cued the animal to either pay attention to the orientation on some trials, or pay attention to the color on other trials. So pay attention to orientation would be one rule, pay attention to color would be the other rule. And this shows a drawing of the monkey's lateral prefrontal cortex. It would be right here on the side of my head, side of my forehead. And this is anterior on the right, posterior on the left, dorsal on top, ventral on the bottom. This is the arcuate sulcus, that's the principal sulcus. Each of the circles represents a location where we had a recording electrode in our array of electrodes. And the colored lines shows where there was increased coherence, increased synchronization of the local field potential signals when the animal was following one rule versus the other. And what you see is exactly what we predicted from mixed selectivity. You see the patterns overlap. There's overlapping between these two patterns, but you can see there's one unique pattern when the animal is following one rule, and another unique pattern, a separate pattern, a different pattern when the animal is following the other rule. So different patterns of synchrony for different rules. We looked at multiple frequencies, everywhere from 1 hertz to 100 hertz, and we found these effects were mainly in the beta range, about 10 to 25 hertz. So we found that beta humming forms ensembles for task rules. Now, since Tim discovered this, We then went and looked at a wide variety of top-down information. We looked at our cats and dogs, numbers, we looked at same versus different. Every time we've taught the animal another top-down high-level concept, we see these beta ensembles, this beta humming forming these ensembles that carry top-down information. Now, with that in mind, let me turn to a related topic, and that is how we control working memory using these beta rhythms. Now, probably all you are familiar with the concept of working memory. Working memory is your sketch pad of your conscious mind. When you say you're holding something in mind, that's working memory. And Michael Lundquist came to the laboratory and he examined data from a task in which we had the animal hold two pictures, which I'm calling S1 and S2, in working memory, in mind. And what Michael did is he did something that was different from what we have done previously. Normally in neurophysiology, we average activity across many trials in order to boost the signal and reduce the noise. And that rests on the assumption that the variability we see from trial to trial is noise, not a real signal. But what if that's not right? What if this variability from trial to trial, the variability in real time, is actually a signal that the brain is using? So Michael examined this activity in real time, and what he found is shown here. So the classic view of working memory is sustained activity. If you're holding something in mind, you have neurons in your prefrontal cortex, other places in your cortex that are continually firing. And that's how you hold things in working memory. But that is an artifact of averaging across multiple trials. If you look in real time, what you see is a very different picture. Neural activity is not continuous during working memory. It's very sparse and bursty. There's bursts of activity followed by silence. Mark Stokes uses this to his activity silent models of working memory. So what's shown here on this figure is time on the x-axis. So here's our two stimuli. The animal sees S1. There's a short gap in time. S2. Then there's a delay where memory delay where the monkey has to hold both pictures in mind. And what's shown here is local field potentials. And on the y-axis is the frequency of local field potential, what frequency they're oscillating at, and in color is shown the oscillatory power. So what we found is that in real time, we see bursts of gamma activity, everything from about 40 hertz or above. We see these bursts of gamma, especially during stimulus presentation, and periodically, once in a while, during the memory delay. The spiking that was carrying the information about the stimuli, S1 and S2, that the monkey was holding in working memory, they all occurred at the time of these gamma bursts. The spikes carrying the spiking carrying the working memories were riding on top of these gamma bursts. And you can see that here plotted on the y-axis is not spike rate, not how often the neurons are firing, but the information that the neurons are carrying. And in red shows the information from spiking that was carried during these gamma bursts. And we see there's lots of, here's information about S1 in one red line. Here's information about S2 in the other red line. We see these neurons, the spiking during the gamma burst, are encoding the stimuli and carrying the working memories during the memory delay. But when we looked at spiking outside of the gamma burst, we found no information at all. So the spiking carrying stimulus information was uniquely associated with these gamma bursts, both in space, both brain space and in time. But the other thing we notice is that these gamma and spiking events alternated with bursts of beta. And you can see that here. Here's now bursts of LFP power in the beta range. These beta bursts tended to happen whenever the gamma bursts and spiking were not happening. So what we found is that beta and gamma are anti-correlated. Here's now the plot of gamma bursting as a function of time. Burst rate is on the y-axis. The line on the top shows the rate of gamma bursting. And what you see for the gamma bursting is exactly what we've seen for many decades if you look at spiking activity alone. There's a burst of gamma and spiking when the stimuli are presented, S1 and S2. And there's this sustained spiking, or this, again, average sustained spiking during the memory delay period that tends to ramp up as the end of the delay nears, which is exactly what people observe for spiking for many, many, many years. But now if you look at the beta bursting is like a mirror image of the gamma bursting. Whenever gamma goes up, beta goes down. Here in the memory delay, beta goes up at first and gamma goes down. Then beta drops down during the memory delay and gamma ramps up. So beta and gamma are anti-correlated. Well, if gamma and the spiking associated with gamma is actually carrying the contents of working memory, what does beta do? Well, I hope you remember that we found that beta is carrying top-down information. They're carrying the rules the animal needs to solve this task. They're carrying information, the knowledge the animal has about how to perform the task. So what we think, our hypothesis is that there's top-down control of working memory by these beta rhythms, which regulate the gamma rhythms. The idea is that top-down beta patterns, these bursts of beta, they're the control patterns, and what they do is they have an inhibitory relationship to gamma and spiking. Whenever beta goes up, gamma goes down. Whenever you want gamma to go up, you want more spiking, more gamma, you drop beta down. The idea is that beta has an inhibitory or gating role to gamma and spiking. And we recently published a paper just a few months ago where we showed how these dynamics could actually allow the brain to perform computation, a concept we called spatial computing. The idea is that these brain applies these rhythms, these beta rhythms, to physical patches of cortex to selectively control the right neurons to do the right things at the right time. Okay. Now, I mentioned the Miller and Cohn model where back in 2000 we wrote this paper showing that the prefrontal cortex has this orchestra score, or roadmap, if you will, and it uses that roadmap to make control activity in the rest of the cortex, kind of like a traffic cop or an orchestra conductor. We used to think that was all done by spiking activity going from the prefrontal cortex to the rest of the brain, rest of the cortex. But now we think that it's all taking place by use of these, via these oscillatory dynamics. Namely that beta is more like a photographic negative of where you want higher cortical activity. You use beta to suppress everywhere where you don't want gamma and spiking to be. And then the gamma and spiking occurs in places where the beta is not, where beta has not landed. Now there's more to this spatial computing story than that. I invite you to read the paper if you want to know more details. Now, when Andre Bastos, who's now Professor at Vanderbilt, and Roman Lunas, when he's now a medical doctor, when they were in the lab, they decided they wanted to look a little more closely at these beta and gamma interactions. So what they did is they use so-called laminar electrodes that have multiple recording contacts along the shaft. Oops. And what this allowed them to do is record from all layers of cortex simultaneously. Your cortex is like an onion. It has 6 layers. Now, why did they care about the layers? Well, we cared about the layers because the superficial layers of cortex, everything above layer 4, are the feed-forward layers of cortex. Anatomically, we know this. They carry information, the superficial layers carry information, sensory information, from the back of the brain, from sensory cortex, up to the front of your brain, your executive cortex. That's taking place in the feed-forward superficial layers. At the same time, we know from anatomy that the deep layers of cortex, they carry feedback signals from the front of your brain, your executive brain, and carry feedback signals, control signals, back to sensory cortex. So the hypothesis was that gamma should be stronger in the superficial layers of cortex because gamma is carrying this bottom-up sensory information, including what the animal has to hold in working memory, whereas the control signals, if they're being carried in the deep layers of cortex, the feedback layers, they should show stronger beta, because we think beta is what's carrying the control signals. And that's exactly what they found. So they did a whole bunch of analyses, including Granger causality analysis, and what they found is summarized in this slide. we found that both beta and gamma were both in superficial layers of cortex, but beta was stronger in deep layers of cortex, and gamma was stronger in superficial layers of cortex, consistent with our hypothesis that beta is carrying top-down information and gamma is carrying bottom-up or sensory information. And what they found is that top-down information, the control signals, were indeed carried by deep layer beta. Deep layer beta, in turn, regulated the weaker beta that was in the superficial layers. And the superficial layer beta inhibited or gated the gamma and spiking carrying the bottom-up information. So whenever beta went down, there was more gamma and more spiking. Whenever beta went up, there was less gamma and less spiking. And that's how we get top-down control of working memory. That's how this is the mechanism, the motif, the functional circuit that allows you to think whatever thoughts you want to think. That think about you can choose what you're going to hold in working memory. You can manipulate those thoughts in working memory. Working memory is under top-down volitional control, and we think this cortical, deep layer, superficial layers interactions are what's producing the top-down control, are what allows the top-down control. But in conducting these studies, we thought, well, why should this just be working memory and frontal cortex? You see this beta-gamma push-pull, this anti-correlation in beta-gamma. You see it everywhere in cortex. If you go back to visual cortex, sensory cortex, the frequencies drop down a little bit towards the alpha range, which is just below beta. It's about, alpha is about 10 to 15 hertz or so, depending on who's defining it. These beta-gamma interactions are everywhere. So both Michael Lundquist and Andre Bastos did a study where they looked all across cortex. And more recently, Diego Mendoza-Holiday and Alex Major from my laboratory looked at data from laminar recordings from 14 cortical areas. And they found the same deep layer beta, superficial layer gamma, push-pull, anti-correlation everywhere in cortex they looked. Whether it was motor cortex, premotor cortex, sensory cortex, prefrontal cortex, it was everywhere. So this can't be, these beta-gamma interactions can't just be working memory and the frontal cortex. It must be something that's general to cortical function. So we thought, well, we wanted to turn to a function that was a more basic general cortical function. And what we turned to is predictive coding. Now, predictive coding, the brain uses predictive coding to regulate sensory information. Your brain can't possibly process all the sensory information flooding into it right now. It would be overwhelmed. In fact, that's what we think happens in autism, is that the sensory signals are overwhelming the cortex and you get sensory overload. But normally the brain prevents this by predictive coding. The brain is constantly generating predictions of the sensory inputs expected in the immediate future. Predicted inputs that the brain could predict, are not informative. The brain saw them coming. That doesn't give the brain any new information. So they're suppressed and not fully processed. However, unexpected inputs, so-called prediction errors, are really informative because the brain could not predict them. That's either new things that the brain didn't expect or any change in the environment. And they're fed forward to update our mental model of the world. Now, virtually everybody agrees that your brain does predictive coding. How your brain does it is not clear. And one common way of thinking about it is that at every level of cortex, there are these comparison circuits. Incoming sensory inputs are compared to the predictions, and if there's a mismatch, that prediction error gets fed forward to the next layer, where there's another prediction, and so on and so on. Now, there's not a lot of evidence for these prediction circuits in cortex. So we thought there's a more simpler, more parsimonious explanation, something we call predictive routing. Namely, that top-down beta is feeding back predictions from the frontal cortex, from the higher cortex back to sensory cortex. And beta suppresses the bottom-up gamma spiking that would otherwise feed forward predicted stimuli. So wherever beta lands in sensory cortex, it's suppressing gamma and spiking there. And those parts of cortex are the parts of cortex that would respond to a predicted stimulus, but they're not responding to a predicted stimulus because beta is suppressing them. And what that means, prediction errors, unpredicted stimuli, they're the places that beta is not landing in cortex. They're processed by circuits in sensory cortex that are unaffected by beta, that are not targeted by beta. So prediction errors are simply normal gamma spiking responding in sensory cortex, in parts of sensory cortex that are unaffected by the beta rhythms. So we tested this recently in collaboration with Nancy Capel's lab at Boston University. And again, the prediction is that the hypothesis is that predictions are fed back by beta, and now in sensory cortex, it drops down a little bit to the alpha range. Again, here's beta 10 to 30 hertz, alpha is 8 to 14 hertz, gamma is 30 hertz and above. And what this shows, we recorded from 5 different cortical areas. Here's the prefrontal cortex and frontal eye fields, frontal cortex. 7A and LIP are parietal cortex, and V4 is visual cortex, sensory cortex. What the colored line shows, the animal was either viewing objects that were highly predictable or unpredictable. And the red lines show stronger coherence in LFPs within and between cortical areas when the animal was viewing unpredicted objects, prediction errors. And the blue shows stronger coherence when the animal could predict the object. And what you can see, it's not perfectly lined up, not perfectly differentiated, but you can see that that whenever the animal was viewing predicted stimuli, there was stronger, excuse me, which is in blue, there was stronger coherence in the alpha beta range within and between cortical areas. Alpha beta coherence dominated in cortex when the animal was viewing predicted objects, which is consistent with the beta and alpha carrying these predictions. Now, area 7A was an exception because Nancy Capel's work suggests that beta and 7A may have a role in updating working memory. But whenever the animal was viewing a stimulus that was not predicted, a prediction error, gamma dominates in cortex, which is consistent with gamma feeding forward sensory information, carrying prediction errors. Now, what was curious, we looked at Granger's causality measures, and also consistent with the hypothesis was that gamma coherence was stronger in the feed forward direction, from sensory cortex to higher cortex. And that's consistent with the gamma feeding forward prediction errors. By contrast, the alpha beta coherence, which we think is carrying predictions, it was stronger in the feedback direction, from higher cortex back down to sensory cortex, from anterior cortex to posterior cortex, consistent with alpha beta carrying predictions. And furthermore, these effects were stimulus specific. We saw these effects wherever spiking preferred that particular specific stimulus, object, or unpredicted object. In other words, these alpha beta signals were targeting specific representations in sensory cortex, representations of that specific predictable object. Okay. Now, last thing I'll tell you about is up at the beginning of my little section on working memory, I mentioned that if you look at activity in real time, you see very complex, sparse activity, like these bursts of gamma and beta with periods of silence, so-called activity silent working memory. But after I said that, I started using all trial average data. But we have begun to look more closely at these dynamics to see what's happening in real time, what sort of dynamics are happening in real time that could explain working memory and high-level cognition. So what we found was that these sparse oscillatory bursts of activity in prefrontal cortex form traveling waves. Now, I've talked about oscillations throughout this talk, and oscillations, important to keep in mind, oscillations, they're not just standing waves where they just, neurons just oscillate up and down in place. These oscillations move throughout cortex. They travel across cortex in waves, like waves of an ocean. So here's an example of this is a real-time recording from an array in the lateral prefrontal cortex. And this is each square has a different electrode in our array. And this is the array was a grid laid down the cortex. The color is the voltage signals. And you can see these oscillatory dynamics don't just make the whole array just go up and down in an oscillation. They're actually moving around cortex. In fact, we know from analysis that this wave is actually rotating around cortex. You can kind of see the rotation here. So why traveling waves? Why are traveling waves so important? Well, traveling waves are super helpful. They buffer information about time and recent network activity. Just the fact that traveling waves exist, they carry information about how much time has elapsed and what the recent activity was in the network. And the way to think about that, if I take a video of a pond and I drop 4 pebbles in the pond one at a time, I could then show you a still photograph from that pond, from that video, and you could tell me how many pebbles have dropped, when they dropped, how long ago they dropped. And that information about recent activity and elapsed time is critical for neural computation. And you get it just because traveling waves exist. Further, we found these traveling waves, they don't just bounce around cortex randomly like billiard balls or ping pong balls or something. They're not just bouncing around in a random way like popcorn or something. They actually move in very specific anatomical pathways. So here's one of our subjects now, one of our monkeys performing a working memory task. In this case, this is posterior on the right, anterior on the left, dorsal on top, ventral on the bottom. So the monkey would be facing towards the left here. And this is, again, it's lateral prefrontal cortex. And what we found is that there was these rotating waves when the monkey was performing the task, and these rotating waves moved in very specific pathways along this anterior anterior posterior dorsal ventral axis. So the fact that these traveling waves are moving in very specific ways kind of tells you that they must be doing something important for brain function. Otherwise, they're not just behaving randomly. And we found these rotating waves actually, they change with different cognitive demands. They change with task performance and cognitive demands. So here's again, now I've plotted here the waves for, we arbitrarily designated these waves as in this subject, the waves were going back and forth in 2 different directions. We called one rotation positive, the other rotation negative. And here's what happened when this animal was performing the working memory task. This is time on the x-axis. This is more waves or less waves, more being up and less being down. The red is the positive waves, the waves moving from dorsal to ventral. And the blue is the negative waves, the waves moving from ventral to dorsal. When the animal was waiting to start the task, there were about an equal number of positive and negative rotation waves, both moving back and forth more or less equally. The moment the animal starts the task, one wave pattern, the negative waves drop out, and the positive rotations, the dorsal to ventral waves, they dominate the cortex. They dominate the prefrontal cortex, and they stay that way throughout the stimulus encoding period, during the memory delay period. Then the moment the animal makes its response and the trial is over with, it goes back to the waves moving in both directions. So we're now working on computational modeling to figure out what role these waves are playing in performance of the working memory task. But the fact they move in very specific ways and they change direction, they change what they're doing with cognitive demand, tells you they must be doing something important. And curiously enough, we found these effects mainly in the beta range, the frequency range that we associate with top-down control. Now, finally, one question I often get asked is if neural activity in the cortex is sparse, if there's lots of this activity silent dynamics, and there's these moving and rotating waves, how do you hold working memories? How do you hold things in mind when there's all these silent periods where neurons are not really firing or doing much? Well, Leo Kotsakov and Adam Eisen in my laboratory, they have been studying this by building neural network models, computational models that have short-term synaptic plasticity. This is a short-term potentiation of synaptic weights that lasts about 750 milliseconds or about 1 second or so. And the idea is that spiking, and this has been shown in the brain, they exist in the brain, and they've been using this property to build computational models, the idea is that the spiking leaves an impression in the network that carries the information between spikes by virtue of this short-term synaptic plasticity. Now, they built models of this short-term synaptic plasticity, and it worked. They were working memory models that could hold working memories. That's not surprising. They designed the model that way. But what they found is there was all these extra benefits from the short-term synaptic plasticity that were not built into the model, but nonetheless made networks work better. They found that networks with short-term synaptic plasticity were more robust to noise. If you degrade the sensory input by adding noise, the neurons with short-term synaptic plasticity, they work perfectly fine with more noise. Whereas if you build a model without short-term synaptic plasticity, where the neurons are continually firing persistent activity, and they're showing these persistent, what's called attractor dynamics, they quickly degrade if you add even a little bit of noise to the input. It's also more robust to degradation of synapses. One standard test of whether a neural network model is working well is it has to be robust to degradation. If you lose some synapses, the network can still work. Well, if you take a model without short-term synaptic plasticity, the traditional model where it's all sustained persistent spiking, again, attractor dynamics, if you take that standard, that traditional model working memory, if you delete as few as 10% of the synapses, the model falls apart. But you add in the short-term synaptic plasticity, and now you can take away as many as 40% of the synapses, and the model still works just fine. So it shows very robust degradation, very robust degradation. And finally, the short-term synaptic plasticity helps stabilize networks, so neural activity doesn't run wild and you get epileptic type activity. Models with based on persistent activity are very prone to that kind of runaway, wild epileptic activity. But you add in short-term synaptic plasticity, and the models are quite stable. So in summary, I want to argue that cognition is an emergent property, not a property of the brain's individual parts and individual neurons, but a property of neurons working together to form these more complex dynamics. The neural activity underlying cognition is not smooth or steady state. It's complex, dynamic, and rhythmic. There are emergent properties like traveling waves that provide new insights into mechanisms and function. And the more specific take-home message is that lower frequency alpha beta waves are the control signals. They control the higher frequency gamma waves and spiking that carry the contents of thoughts, sensory information, perceptions, memories, etc. And we think this interplay between alpha beta waves and gamma waves are taking place in the superficial and deep layers of cortex. And we think this is a general cortical motif that plays a general role in regulating cortical function. Not just working memory, but attention, predictive coding. Virtually anything your cortex does will depend on achieving the proper balance between these top-down beta waves and these bottom-up gamma waves. So I thank you for your attention. I thank you for the opportunity to speak to you, and most of all, I thank my students who do all the hard work in conducting these studies. Thank you.
Hello. Sir, we'll take up a couple of questions from the audience. So
Sure.
anybody has questions, you can ask.
So thank you for your thought. I'm going to ask you about the analogy that you talked about red and blue nodes. So you said that if mixed selectivity neurons are there, then we can encode information of a mixture of colors between red and blue dots.
Mhm.
But we can only do that because the concept of red and blue dot is already present. You are already giving the concept of red and blue dots. But if we want to encode a new concept, how are mixed selectivity neurons recruited into creating a new concept, encoding a new concept?
Right, that's it. Sorry, go ahead.
And another thing, you said by a turn of the usual phrase, neurons that fire together, wire together. You said neurons that hum together, wire together. You said
Yep.
that if we think one thought, then a network of those mixed selectivity neurons get activated. But that thought requires the creation of that network. So isn't that a tautological or a circular argument? I mean, if you want to think the thought, that network of blue thought has to be activated, but that network of blue thought creates the creation of its first. So isn't
Right.
that a circular thing? I
No,
mean, where's
no,
the beginning of that?
the beginning of it is in how your brain gets wired up. That's an excellent question. Excellent question. The beginning of how your brain gets wired up. So I mentioned the road and highway system, the anatomy in your brain. Your brain is wired to respond to certain during development, genetics. Genetics wires your brain to respond in certain ways. Those are the primitives your brain has. And then during early development, sorry, excuse me. During early development, your brain starts laying down new wires. It changes its wiring to respond to statistical regularities in the world. It's building all these primitives of thought, right? And now when you want to, but those are going to be overlapping in anatomy because of this property of mixed selectivity. But now when I want to activate one particular thought, one particular thing, I can combine those primitives through the mixed selectivity neurons and activate a unique network that now corresponds to that thought. Now, it could be something you recognize that's highly familiar, because your brain has these primitives all wired in there. The road and highway system is still very, very, very important. That's the beginning of everything. So you have the potential to activate this information, these familiar concepts and thoughts. But a new concept and thought, well, as my PhD advisor, Charlie Gross, used to say, there's really no new thoughts, there's recombinations of old thoughts. And that's if you take these primitives, and because of these mixed selectivity neurons, if there's no mixed selectivity neurons, you would have these primitives and they'd be separate from one another. They couldn't find one another. You couldn't construct them together. You couldn't pull them together. But now with these mixed selectivity neurons, you have all these primitives corresponding to the basic knowledge, the latent knowledge your brain has. And because of these mixed selectivity neurons, these familiar primitives, can be combined flexibly into a brand new thought or concept. That's the idea. You need mixed selectivity to pull together information that in the past hasn't been together before, but you're now combining it anew. And the mixed selectivity neurons allow you the flexibility to do that.
Hello.
Hello?
Good evening, Professor.
Hello?
I have a question with relation to the synaptic plasticity.
Yep.
We have studied about persistence of vision in the human eye, where if multiple images overlap over for a second of time, then the human brain cannot interpret the difference between the two visions. And that is because the image is imprinted on the retina for some time at the receptor level. So that is considered as a defect of the nervous system.
Yep.
And today, when we talk about synaptic plasticity, it is this retention of the signal in the neural network that is helping us to maintain the conscious thought, the working thought.
Yep.
So are both of these cases the same, or is there a positive or negative in each?
No, I think they are. I mean, like your brain develops mechanisms to do certain things, but they're not perfect mechanisms. So in this case, there's two things going on with the kind of phenomenon you're describing, is that when neurons fire, they'll naturally fire they don't turn off instantly. They have a little bit of additional firing. Then you also have the short-term synaptic plasticity. And most of the time that works pretty well and allows our brain to do things like hold things in working memory without having activity constantly running in your brain. So it's energy efficient. But occasionally in situations like you described, you can actually, overload the brain and you get this essentially confused representations, because the synaptic plasticity that works well in most cases is now not working very well in that particular case. We have the overlapping activity. But that's the idea exactly. That the short-term synaptic plasticity makes your brain work better most of the time, but it's not perfect. So there's other situations where it actually works against your brain, your brain function, like in the situation you described where images are overlapping.
Thank you, sir.
Hi,
Any more questions?
Yeah, Professor, for the nice lecture. Thank you very much.
thank you.
So I have a small query.
Mhm.
As you mentioned in one of the earlier slides, while you were talking about mixed selectivity neurons, where the increases the storage capacity.
Yep.
So do you mean the increase in capacity is physical or is it
Oh yeah,
virtual?
I do mean that. So what Stefano Fusi and Matteo Rigata did is they built a computational model that had mixed selectivity neurons in it, and then a computational model that was more traditional, where every neuron had one function only. Now, neural activity in the brain is highly correlated. I talked about the oscillations. Those are millions of neurons all oscillating together. So now you can imagine if I have a million neurons in the brain and they all behave identically, you might as well just have one neuron. So the fact that neurons are highly correlated, many neurons are highly correlated, means there's going to be a natural limitation how much information can be distributed those neurons can store. And what they found is that if you build a traditional model with every neuron having one function, that model, now mind you, this is a model, it's not the whole brain, it's a simpler representation of the brain, but that model can only learn about 3 or 4 tasks, and then you've reached the storage capacity of that model. But now you take that model and you start adding these mixed selectivity neurons, and once the mixed selectivity neurons are there, all of a sudden that capacity limitation is gone, and that model can learn hundreds or even thousands of tasks without saturating. And it's because the information is not just limited to a small number of correlated neurons, because the information is highly distributed across many, many neurons. So you're distributing the memory across so many neurons that the neurons are only, you only use up a little storage capacity of any individual neuron. So now you have a network that can store virtually an unlimited amount of information.
Or does it mean the storage capacity is fixed, but how we are being trained to store more knowledge and more information?
Sorry, I didn't understand.
So are we trying to store more information by training?
Yeah, that's what you teach the model one task and you teach it more tasks. And I mean, our brains are constantly learning new things over a lifetime. And yet the brain has the capacity to keep, I mean, I could store a lifetime information experience in my head in latent format. So there's got to be some mechanism that allows the brain to have this very, very large storage capacity. And this mixed selectivity neurons seem to be part of the answer because they allow information to be distributed across many, many neurons, which effectively increases the storage capacity of the brain.
Okay, yeah, thank you.
Thank you.
Okay. Professor Miller, yeah, very beautiful lecture. I think your lecture has excited a lot of questions. A very short question. Does this research, can we exactly pinpoint what is thought, not content of the thought?
Wow, that's a difficult question. What is thought? I would say thought. If you want my opinion about what consciousness, if you talk about conscious thought, my opinion is that consciousness must depend to some extent or another on these oscillatory dynamics, these brain rhythms, because consciousness is a unified experience. I'm sitting here in my home office, and I have this sights and sounds around me. I have the ambient feeling of the temperature. I have the feeling of the chair on my body. My conscious experience are all these things all linked together into one unified experience. So somehow consciousness must depend on getting large bits of your cortex all doing the same thing, all on the same page, all combining synchronizing their activity so that we get this unified experience of consciousness. So I think conscious thought must depend to some degree or another on these synchronized activity across cortex, and that's what these oscillatory dynamics are evolved for.
Okay, great. Thank you.
Hello, Professor.
Hello, hi.
Hi. We talked about the control signals, alpha and beta.
Mhm.
Is there any relationship between these control signals
and the intention. Before we do any sensory activities, sometimes I have an intention. For example, to lift in a hand.
Yeah.
Is there any
That's it.
relationship between them?
Oh yeah, we think it's exactly what these top-down controls, the alpha beta dynamics are doing. They're allowing things to happen in the brain. For example, a classic study from motor cortex from many years ago. If you have a subject, you tell a human subject, you say, give him a button, and say, I want you to press the button, but don't do it yet. Wait till I say go. Wait until I give you the go signal. And the whole time the person's finger is hovering above the button, you get lots of beta in motor cortex. Then the moment you give them the go signal, beta drops down, gamma goes up, the motor cortex issues a command, and the person presses the button. Okay, we've known that that's how motor cortex works for a very long time. Now, where did cognition come from? Cognition evolved from the neural mechanisms for voluntary motor control. So it's not a surprise to me that cognition would work by the same alpha, beta, gamma dynamics that your motor cortex works at, because that's where cognition came from. That's where it evolved from.
Following question.
Yep.
Can you give some idea about what is the origin of this intention from so far study, whatever we understand brain so far?
Yeah, I mean, like that's one of the toughest questions in neuroscience. If I had the answer to that, we could all go home and stop working on the brain. But I will say that we think that in our original Miller and Cohn model from 2001, we thought that what the prefrontal cortex is doing, in the end, the brain's got to represent information. Everything must work by representation. So what the prefrontal cortex is doing is forming these representations that reflect the logic of a goal-directed task, what the animal needs to do in order to engage in this goal-directed behavior. So that must be a pattern of activity in the prefrontal cortex. And with Miller and Cohn, what we said back 20 years ago or so, 22 years ago or so, is that these representations of the prefrontal cortex aren't just the esoteric representation of the logic of a task or the goal-directed information. The representations are in the form of a roadmap that the rest of the cortex needs in order to engage to engage in that behavior. You have this roadmap, you have this representation of the prefrontal cortex that represents the volitional top-down act you're involved in. And that representation is also a map of which circuits need to be activated in the rest of the cortex to make that thing happen. So in the end, the intention, volition must be a representation problem. Must depend on the brain developing these representations that reflect the goal and the action needed to get to that goal or sub-goal, and the action needed to get to it. And we think it's got to be represented in terms of which neural circuits need to be activated, because that's what your brain needs to do to engage in that intention.
Thank you very much, sir.
Thank you.
I think we have no more questions left. Okay, so I'm assuming we have come to the end. I would like to thank you on the behalf of Bhaktivedanta Institute and the audience. I extend my heartfelt gratitude to Professor Earl K. Miller for sharing with us his deep and vast research knowledge on working memory and related subjects. It was truly a highly invigorating presentation and a wonderful session. Please give a wonderful applause for Professor. Thank you so much, sir. Thank you.
Thank you very much.